Bismillahirrahmanirrahim. Topic liver. Liver is the most important and largest organ, largest on the basis of its function. On the basis of size, the largest organ of the human body is skin. Location of the liver. It acquires central position of the abdomen and is involved in almost every aspect of body's physiological activities. Because of its central role, liver diseases can be extremely life-threatening. The liver has multitude of important and complex functions. Multitude mean large number, great number or various. Some of these functions are carbohydrate metabolism, fat metabolism, proteins metabolism, detoxification, storage and bile. The carbohydrate, fats and proteins, they can be anabolized or catabolized in the liver depends upon the need of the body. First, carbohydrate metabolism. Glucose is a vital energy source for cells and its level in the bloodstream must remain constant. The liver helps maintaining blood glucose in response to the pancreatic hormones insulin and glucagon. After a meal, glucose enters the liver and level of blood glucose rises. When there are many numbers of glucose units in the body, the process occur in the liver is known as glycogenesis. Glyco is for glycogen, genesis is for formation or synthesis. When many number of glucose are present in the body, these glucose units join together to form glycogen in response to hormone insulin. If the body again need energy, the process occur is known as glycogenolysis. Combination of two words, glycogen and lysis. Lysis mean breakdown. The glycogen will again convert back into many units of glucose in response to the hormone glucagon. Third, gluconeogenesis. Gluco word is for the glucose, neo word is for new and genesis word is for synthesis or formation. In this process, amino acids are converted to the glucose. The pathway that shows the conversion of amino acid to sugar. Cells involved, muscle cell, and this is the liver cell, that is hepatocyte. In the muscle cell, whenever the energy is needed, glucose will undergo glycolysis, results in the release of ATPs. At the end of glycolysis, pyruvate are produced. If no more energy is required, this pyruvate can convert it into amino acid alanine. Alanine by means of blood will move into the liver cell or hepatocyte. If the energy is required, the alanine will convert back into pyruvate and pyruvate will again convert back into the glucose. The process is known as gluconeogenesis. Again, glucose can move to the muscle cell by means of blood. Second function, fat metabolism. If fats are in excess in the body, liver prepare it for storage in the subcutaneous tissue. Sub word is for below or under, cutaneous word is for the skin, that is adipose tissue, and other storage depots. Depots mean deposits where they accumulate, fat accumulates, like bone marrow, visceral fat. What is viscera? The internal organs in the main cavities of the body, especially those in the abdomen. Fats can be anabolized or catabolized. The formation of fats is known as lipogenesis. Lipo word is for lipid. Genesis mean formation or synthesis. Lipids can be catabolized. The process known as lipolysis. Lipo word is for the lipids. Lysis word is for the breakdown. The process of lipolysis and lipogenesis. If the body needs energy, the stored triglyceride can break down into glycerol and fatty acid. The glycerol can be converted into PGAL that undergo the process of glycolysis, and fatty acid by means of oxidation can produce many acetyl groups. This acetyl group, when combined with the coenzyme A, results in the formation of acetyl coenzyme A. Now, if the body needs more energy, this acetyl coenzyme A, from the acetyl coenzyme A, acetyl groups combine together to form fatty acid. PgAl can convert back into the glycerol. This glycerol and fatty acid combine together to form triglyceride. So this process is the lipogenesis, while this process is the lipolysis. Third function, protein metabolism. The amino acid can be converted into fats. If many numbers of amino acid are present, they can be converted into fats. This is the amino acid conversion to fat. Amino acid isoleucine can be converted into a compound known as alpha keto beta methyl valerate, which can be converted into 2-methyl butyryl coenzyme A, which finally converted into fatty acid. So the conversion of amino acid to the fatty acid or fats. Second way of protein metabolism, gluconeogenesis, that is amino acid conversion to the glucose. The diagram of amino acid conversion to the glucose. Amino acid, they can be converted into pyruvate, which can convert into glucose in the liver cell hepatocyte, the process known as gluconeogenesis. Third, deamination, removal of amino group from the amino acid in the form of ammonia or ammonium. Deamination, amino acid glutamate, when it combined with the water in the presence of oxidized form of NAD. Products produced are NADH2, alpha ketoglutarate, and ammonium ions are released. Fourth function is detoxification, means removal of toxins. Detoxification and destruction of endogenous and exogenous harmful substances. Endogenous, that is produced inside the body, exogenous, that is coming from the outside body, from outside into the body. Livers on phagocytes are known as the Kupffer cells. They reside or are present within the lobule. This is the structure of lobule. Liver is composed of many lobules. Lobule itself composed of many components, like this is the bile duct, hepatic portal vein, hepatic artery. These structures are the sinusoid. The function of sinusoid is it serves the location for mixing oxygen rich blood from the hepatic artery and nutrient rich blood from the portal vein. Then the central vein or hepatic vein. These are the cells of the liver known as hepatocyte, and in the sinusoid these yellow color structures are the Kupffer cell or phagocytes. Functions: phagocyte digest and destroy cellular debris and invading bacteria. Invade means that gets entry into the body. Other exogenous substances such as drugs and alcohol are also detoxified. How the drugs are detoxified? The drugs are converted from the fat soluble form to the water soluble form, so they can easily be eliminated or removed from the body. Alcohol detoxification. Alcohol in the presence of enzyme alcohol dehydrogenase can be converted into the acetaldehyde. Acetaldehyde in the presence of another enzyme aldehyde dehydrogenase converted into acetate. Acetate will become further broken down into water and carbon dioxide, which will be released from the body. Fourth, amino acids are deaminated in the liver. This is the example of deamination. The glutamate, when it is converted into alpha ketoglutarate, results in the release of ammonium ions. Some hormones, if they are present in excess of the body need, they will become inactivated in the liver, And last, bilirubin, that is the product of old RBC, also detoxified and rendered or made harmless by the liver metabolism. The process of breakdown of old RBC. Old RBC, by the process of phagocytosis, enters into the Kupffer cell. Inside the Kupffer cell, the breakdown of hemoglobin occurs. Hemoglobin, when it breaks down, it will become separated into heme part and globin part. Globin part will again form amino acids, while the heme part will again divides into two parts. Iron will become separated and bilirubin will become separated. This bilirubin results in the formation of bile. Bile from the liver goes into the gallbladder and stored there. The iron will become transported into the bone marrow for the formation of new RBCs. Fifth function, storage. Hepatocyte or liver cell stores some vitamin like vitamin A, B, D, E and K. Hepatocyte can also store some minerals like iron and copper. Glycogen is also stored in the liver as well as in the muscle cells. And last function of the liver, bile. Composition of the bile, it is composed of water, cholesterol, phospholipids, bilirubin, electrolytes, that is sodium, calcium, potassium, magnesium, phosphorus or chloride, and salts. The bile is produced in the liver, but it is stored and concentrated in the gallbladder. The presence of fats in the gut during meals stimulate the gallbladder to empty or to release the bile. Bile enters the duodenum, first part of the small intestine. The function of which is they emulsify the fats into smaller globules. Emulsification. Definition. Emulsification is the process of dispersing one immiscible liquid in another immiscible liquid. Immiscible mean that cannot or do not mix with one another. If you put a drop of oil on the surface of water, you can see these drop-like structures. This process is known as emulsification. So due to emulsification, fats are broken down into smaller globules. These globules can be then easily broken down by enzyme lipase. Metabolic wastes, drug products, bilirubin, they are excreted in feces as a waste material.